Welcome to the lecture series in food microbiology. In the last couple of sessions, we have been handling the different types of food products, the plant and the plant products, meat from animals, egg from poultry, then freshwater and marine fish and fish products. We have been seeing what are the predominant type of microbes which grow in them and what are the conditions in which we should store them to prevent them from getting contaminated by microbial growth and also to prevent spoilage happen to them. In today's session, I am going to talk about crustaceans. These are also marine organisms which are predominantly used in food. Here in this case, we have three categories which are very well used as food products. One is the shrimp, that of the crab and lobster. Now, crustaceans are protected because of the outer shell which they have. But the point is, when we talk about these animals, when we compare them, the shrimp versus crab and the lobsters, the shrimps have much more microbial spoilage than when compared to crab and lobster. The reason for this is the moment you harvest the shrimp, it dies. But crabs and lobsters are alive until they are processed, and the shrimps die during harvest. The second aspect is when we grow these, like you have shrimp farms, you have the crab farms and the lobster farms where the animals are grown, where the crustaceans are grown as food, for export or for import, whichever is for local usage or for export. So here, depending upon the conditions in which they are grown, we have the bacterial diseases happening them to kill these shrimps in a much more. For example, you have the white spot disease of the shrimps, which can cause huge damage to the shrimp farmer. And the second case is the nature of these. So shrimp has a potential to get spoiled much more than a crab and a lobster. Now, the flesh of crustaceans are generally rich in the NPN compounds. Amino acids, they are extremely amino acid rich, and among the amino acids, what you are going to see much more in a high combination is arginine and trimethylamine oxide, and it contains 5% glycogen and a pH above 6. Now, this condition, the richness of the NPN compounds and 5% glycogen and the 6 pH normally will promote the growth of Pseudomonas and many other gram-negative rods. So, the predominant spoilage organism in this case is going to be Pseudomonas mainly. Now, when you have microbial growth here in the shrimp, the first evidence is by order change due to the production of the volatile metabolites of the NPN compounds. That is, these amino acids as well as trimethylamine will get hydrolyzed, you will get the decay of them. So, the decay and putrefaction will cause a volatile metabolite, which is in turn you will see a bad odor because of that. The second aspect is slime production and the loss of texture and color. So, the texture becomes lost, the fat becomes a sort, the structure of the flesh becomes lame or change, the color changes, you have slime on the surface, slime production on the surface along with an accompanying off odor. All these indicates that the harvested shrimp has been damaged, it has microbial growth. But if the moment it is harvested, it is processed and then frozen immediately, you can minimize the loss, but you cannot completely rule out microbial growth altogether. As compared to fish and crustaceans, oyster, clam and scallop meats are also lower in NPN compounds but higher in carbohydrates. Here the glycogen is going to be 3.5 to 5.5% with pH normally above 6%. But in all these oysters, clams and scallops, the outer molluscan shell normally will help to protect the animal, and it is normally taken out, harvested and immediately processed. So, you have lesser chance of microbial contamination here when compared to shrimp concerned. And the mollusks are normally kept alive until processed. This we call this a shucked. Thus, the microbial spoilage occurs only after processing, not during processing. The resident microflora are predominantly Pseudomonas and several other gram-negative rods. And during refrigerated storage, the microorganisms metabolize both NPN compounds and carbohydrates. So what happens here is, in the case of molluscans, since they are kept alive till they get processed, after processing, it is during the storage that you are going to see evidences of microbiota growing. So which is going to be the growth of pseudomonas and other gram-negative rods. So when you refrigerate and store, if the microorganisms are present in them, they will utilize the NPN compounds which are present in the meat along with the carbohydrates, and the utilization of these in turn will produce the slime, bad odor, and the other accompanying changes to the stored food concerned. The breakdown of the nitrogenous compounds mainly by Pseudomonas and Vibrio, especially at refrigerated temperature, results in the production of ammonia, amines, and volatile fatty acids. And in the case of fish, we have a lot of microorganisms which are pathogens associated with fish. We have the Vibrio cholerae, the Vibrio parahemolyticus, Vibrio fulmicus, Clostridium botulinum type E, the enteric viruses, the scromboid fish poisoning, the paralytic shell poisoning. All these foodborne illnesses and pathogens are associated with fish. The next category which we are talking about is going to be canned foods. In storage of food by canning, normally after transferring the food to the can and sealing it, normally the can undergoes heat treatment. You seal the can and then you make it undergo heat treatment so that microorganisms get killed. And this extent of heat treatment is predominantly dependent on the pH of the food. So what is the nature of the food concerned? How are you storing it? For example, you have canned vegetables, you have canned fish, you have canned oysters, you have so many different stuff. You can store it in normal saline water, that is brine, or you can store it in tomato sauce or in some other components. So depending upon the pH of the food concerned and what is the storage medium, then the heat treatment which the can undergoes will change, because you don't want to cause changes to the storage media concerned there in the can. If high pH, for example 4.6 and above, which is also called as the low acid food, they are heated to destroy most of the heat resistant spores of the pathogenic bacteria. For example, Clostridium botulinum, to ensure that the product is free from any pathogen. So at low pH, that is low acid, that is a high pH in an acidic level canned food, you can heat them without any problem happening or any changes happening to the food concerned. So this heat treatment in turn will also destroy even the spores. Especially there you have the chances of Clostridium botulinum spores growing there, and as you know, this is a lethal food poison. So heat treatment given here can kill even the spores of the Clostridium botulinum, so the product will be free of any pathogen as long as the seal is intact and it is not broken open. The spores of the spoilage bacteria, which have greater heat resistance than those spores of Clostridium botulinum, can survive. So you do have some spoilage bacteria whose spores can withstand higher temperatures rather than even more than what Clostridium botulinum comes. Botulinum we are bothered because it's a toxin, but you do have other spore formers which might not be a pathogen but which can be a spoilage organism, the spores of those which can grow in a canned heat treated food concerned. So these products which are called as commercially sterile, so you need to look there whether is it like normally sterilized or is it commercially sterile. The term commercial sterilization indicates that the product is free from not only microbes but also from even the spores of the pathogenic or non-pathogenic spoilage organisms. The spores that survive the heat treatment designed to destroy Clostridium botulinum are thermophilic spores, and these can germinate at 43 degrees and above, and once germinated some can grow at temperatures as low as 30 degrees centigrade. So spores, as you know, are normally dormant, and if they are thermophilic spores, Even when kept and given a heat treatment beyond 43 to 45 degrees centigrade, the spores are still dormant, they are alive. Yeah, metabolically almost in inactive state, but they are still alive. But suppose after the heat treatment, the conditions becomes conducive for the spore to germinate. For example, once the spore germinates, then that product is irreversible. The process is irreversible, it germinates and gives rise to the live microbe. Okay, and this, while you are storing it at 30s, when the organism can grow. So your germinated spore will now proliferate to give the bacterium, which is happily proliferating at 30 degrees. It will reproduce, and your canned product, in spite of having the brand that it is commercially sterilized, will have. The moment you open it, you will see that the product has become spoiled because of the microbial growth. This is because of the thermophilic spore which was inside, which germinated and gave rise to the organism. The other food group, which we designate as low pH or high acid food, is treated all vegetative cells and some spores. The low pH normally will inhibit the germination of spores, and the Clostridium botulinum spores of some acidic thermophilic spoilage bacteria can germinate and grow when the products are stored at higher temperatures, even for a shorter time. Here, the microbial spoilage is due to three main reasons. One is the inadequate cooling after heating, or high temperature storage allowing germination and the growth of thermophilic spore formers. See, after giving heat treatment for a particular time, the can has to be taken out and allowed to cool normally, naturally. You cannot immediately cool it. The sudden temperature fluctuation actually gives a stimulus to the spores to start germination. So, and if it is not cooled properly before getting stored, that also can stimulate the microbial growth. So, inadequate cooling after heating, number one. Two, storing at very high temperatures, because here we are talking about thermophilic organisms. They will proliferate at high temperatures. So, high temperature storage, which will allow the growth of the microbes, the thermophilic spore formers. The second is inadequate heating, resulting in the survival and the growth of mesophilic organisms, the vegetative cells and spores. The heat treatment given to the canned product is not sufficient. So, the temperature was not high enough to kill either the vegetative cells or even the spores. As a result, in spite of the heat treatment, both the vegetative cells as well as the spores grow, germinate, proliferating, spoiling the food concerned. The third is leakage, which can be microscopic in the cans, allowing microbial contamination from outside following heat treatment and their growth. So, suppose there is a small microscopic or a submicroscopic pore because the can was not properly sealed. After heat treatment and after adequate cooling, even if the temperature of storage was normal, preventing microbial growth, the small leakage is enough for the microbes. That small hole is enough, microscopic hole is enough for the microbes to make an entry into the canned food, and that canned food is actually an optimum, a rich medium for nutrient for the growth of the microorganism, thereby allowing it to grow, proliferate and spoil the organism concerned. So, you have three major reasons why canned food get spoiled. One is inadequate cooling after heating or high temperature storage. The second, the heat treatment is not sufficient. The temperature hike is not sufficient to kill either the vegetative cells or even the spores. 3, leak. The seal has not been properly set. So, because of this leak, microbes gain entry and spoil the food concerned. In the last couple of lectures, we have been seeing that depending upon the type of food concerned, the microbial spoilage rate will vary. For example, if we talk about the vegetables and the fruits, the plant products, they formed a separate category. The type of spoilage caused to plant and plant products was totally different. For example, fruits had a different sort of microbial attack. They were more subject to fungi and molds rather than to bacterial attack. You have the vegetables which had a different sort of rot. We saw the rot in them. Then the cereals. In the case of cereals, because of the lesser amount of water activity, normally you did not see much of bacterial growth. But there, the predominant spoilage organisms were fungi. We also saw the storage grains getting, because of the respiration, which could in turn, because of improper sporage conditions, the fungal spores could germinate in them, causing the production of aflatoxins. And these aflatoxins in turn can cause not only harm to the animals consuming, for example, barley, wheat, etc., which are used for fodder for cattle as cattle food, or even in humans. For example, history says that earlier, in the case of during the Industrial Revolution time, along with wheat, people used to add some amount of rye also as for baking bread. And at that time, improper storage conditions actually made the people, and bread was made out of it, people consumed it. And you had people becoming hallucinogenic because of the production of because of the consumption of the aflatoxins, which in turn, how did these aflatoxins come? Because of the improper storage of the grains. They were not properly stored. As a result, the fungal spores grew. Even if you don't see the external evidence of the fungal growth, the aflatoxins secreted by the fungi would still be inside the grains, and if present in a higher amount, can cause toxicity. So that was the sort of changes we saw in vegetables, plants and plant products. In the case of animal, for example, in the meat, we saw that the health of the animal concerned, the hygiene and the sanitation practices which are followed at the slaughterhouse and the processing conditions in turn could influence the microbial growth in them. In the case of egg and egg products, irrespective of pasteurization, freezing and taking precautions, the shelf life could not be increased much because both the albumin as well as the egg yolk protein, they are highly nutritive media for the growth of microbes. So the moment you take it out, either as storage as a liquid food or storage as a long food, is going to be tough in spite of the calcareous outer shell which we talk about. The predominant microorganisms in seafood, whether freshwater fish or in seafood like your molluscans, the crustaceans, etc., the nature of the microbial flora became changed because of the type of food which is present in them. You had much less carbohydrate, but predominantly more amount of what we call as the NPN compounds there. So this in turn, for example, fish has a very high amount of unsaturated fatty acids. So because of the change in the nature of the nutrient composition of these, the predominant microorganisms which cause spoilage there also changed. So as a summary, if you look into the type of microbial spoilage caused to the food concerned, depending upon the different types of the food or the food groups, each differ in the microbial spoilage characteristics. The predominant nutrients being metabolized and the predominant microorganisms growing rapidly under an environment determine the nature of the end products that are produced, which in turn are associated with the spoilage characteristics. So depending upon the spoilage organisms which inhabit, the type of spoilage, whether the texture is getting changed, whether you are going to see a off odor, whether you are going to see a sliminess, whether you are going to have more amount of rancidity, all these changes depends upon the nature of the food and the predominant microorganism we talk about. And an understanding of all these parameters is important in the first hand determination of the causes of spoilage. So, and this in turn, not even the causes of spoilage, but if you are going to talk about products, if you want to make more type of ready to eat products, you want to increase the shelf life of these particular type of products, or you want to mix up, say for example, a seafood with an animal and make increased shelf life. Under such condition, you will have to find out the predominant microorganism, 2, the conditions, optimum conditions for each growth, and 3, the processing in which this type of microbial growth can be completely prevented. Then the precautionary measures you have to take. What would be the predominant amount of shelf life with which you can store this without it getting microbial growth concerned. So all this knowledge becomes extremely essential. All the parameters become important in such a case. Important questions which comes to one's mind when we talk about microbial spoilages in different type of food concerned, when you are packaging, for example, export is one aspect, short-term storage is the second aspect. Depending upon the type of microbes which cause spoilage to these, how will you devise packaging mechanism for different types? So, if you consider the microbial flora, the type, for example, we talk about modified atmosphere packing, vacuum packing, canning, then tins, Then you have the ready-to-eat products. Ready-to-eat products containing a mixture of cereals and vegetables and fruits. Then you have the meat products. So for each of these, when you talk about, you will have to design the optimum packaging mechanism so that you will have the minimum amount of microbial contamination. That is number one. The second aspect is when you are combining different types of products, you also will have to take into account the different types of microbes which can infest, because predominantly you are now mixing up stuff and you now will have an atmosphere conducive for the growth of different types of microorganisms. Some of them would be pathogens, some of them would be non-pathogens. The second point which you have to remember is low water activity. Normally, extremely low water activity will inhibit microbial growth. So, dehydrated products would have a better chance of lesser microbial spoilage and longer shelf life rather when you design a packaging mechanism. So, all these criteria would have to be satisfied when you design an optimum packaging mechanism for each type of food which we talk about. Second, storage. You have designed a good packaging system. You know that in this package the shelf life has been increased, but what is the storage condition? Are you going to keep it in freezer? Is it going to be in refrigerated temperature, or is it going to be at room temperature? In each of these, your packaging mechanism will have to change. The third aspect is, even if you keep it in a freezer or even if it is in a refrigerator, the food stuff should not undergo temperature abuse. If it undergoes temperature abuse, you know that the dormant spores can germinate, or already existing vegetative cells, which might be less in number, can predominantly start growing. So this would be the second aspect you have to take into consideration. The third is, what are the safety issues? Safety issues mainly, I would talk about the pathogenic organisms getting into the food. Spoilage is one aspect, the pathogens in the food is the second aspect. Normally, pathogenic organisms are very less normally. When you compare the normal microbial flora of a particular food concern, it would be the non-pathogens which predominate, not the pathogens. But still, even when very small amount in number, even these small number of pathogens in turn can cause foodborne illnesses, what we call as intoxications, gastroenteric diseases, or even lethality. So, depending upon the concentration of these pathogens, you will have to take precautions. So, storage, when we are talking from a food industry point of view, microbial spoilage causes a huge amount of loss because of spoilage is one aspect. So spoilage organisms causing loss is one aspect. The growth of pathogens in turn causing lethality or even causing illnesses is the second aspect. Both of this has to be prevented when we are talking about a food industry. And in each step, you have not only sanitization and procedures which we take about, but we do have what we call as regulatory mechanisms to check the predominance. Step by step, you have the ISO regulations which also state that for a particular food industry concerned, depending upon whether you are getting depending upon the nature of your food product, plant product, animal product, fish, ready to eat, whatever, the certification and the legal hurdles, the regulatory issues and the safety issues which a food industry has to modify and accept are manifold, especially because microbial spoilage and microbial contamination leading to food poisoning is extremely lethal, which is not acceptable either by the consumers or by the governments concerned. Thank you.